And continuing our talk on rheumatoid arthritis, we're now going to talk about immunologic tolerance and autoimmunity. When we were considering how the enormous diversity of antigen binding sites in antibodies is generated, we noted that, not infrequently, self-reactive antibodies are generated. We also noted that immunologic unresponsiveness to self is essential for optimal function of the immune system and the health of the host. These facts raise the question as to how the adaptive immune system acquires the ability to distinguish between self and non-self. Several different mechanisms are used to create immune tolerance. Both central and peripheral mechanisms are involved. In central tolerance, self-reactive lymphocytes are purged during development. T cells and B cells which bind antigen with high affinity are deleted. But because not all self-reactive T cells are purged as a result of central tolerance, there are additional peripheral mechanisms that include anergy, which is functional inactivation, peripheral deletion, clonal ignorance, and suppression of immune response by Tregs. T cell anergy is induced if CD4 positive T cells recognize antigen in the absence of a co-stimulatory signal, usually provided by an APC. Peripheral deletion is induced when T cells are hyperstimulated by high doses of antigen and high concentrations of interleukin-2, which activate apoptosis. Clonal ignorance refers to the inability of self-reactive lymphocytes to recognize or respond to peripheral antigen if the antigen is sequestered in a protected site. Tregs are important in maintaining peripheral tolerance by producing anti-inflammatory cytokines including TGF-beta, interleukin-10, or interleukin-14 that suppress immune responses. Beta cells require T cell help to become activated. In the absence of T cell interaction, B cells will become anergic. To summarize, these processes are believed to underlie unresponsiveness to self-antigens. So first, there is sequestration of self-antigen in privileged locations such as the brain. 2. Central or peripheral deletion or anergy. And 3. Suppression of responsiveness by regulatory cells. When any of these normal processes are disrupted, autoimmunity may be a consequence. And continuing on, we'll talk about mechanisms of autoimmunity. The research literature on autoimmunity is vast and supports the contention that while there are many hypotheses regarding the etiology of autoimmunity, for now they remain sophisticated speculations. Nevertheless, because this is an area of such importance in medicine, we will attempt a summary of the most widely accepted areas. It is universally agreed that autoimmunity is a multi-step process that begins with a trigger event, such as an infection or response to toxic agents like cigarette smoke or various drugs, e.g. procainamide. One of the best understood mechanisms is so-called molecular mimicry, in which infectious agents are the source of antigens that cross-react with host tissues, triggering activation of autoreactive T or B cells. The prime example of this phenomenon is believed to be rheumatic fever, in which autoantibodies to the M protein of Streptococcus pyogenes cross-reacts with cardiac myosin, laminin, and perhaps neuronal antigens. Another mechanism is the loss of immunologic privilege or release of sequestered antigens. For example, brain and the anterior chamber of the eye are not exposed to the immune system. If they are damaged, antigens can enter the circulation, producing experimental encephalitis or endophthalmitis. Still another mechanism is presentation of previously invisible peptides, which are called cryptic epitopes. During T cell maturation in the thymus, APCs carrying self-antigens are purged by negative selection, cryptic epitopes are usually present in low quantities on APCs because they are processed inefficiently or they possess low affinity for the MHC groove. Those cryptic epitopes present in low concentration cannot delete the autoreactive T cells and thus become part of the peripheral T cell army. Chemical alteration of proteins by inflammation, drug exposure, or biochemical processes associated with aging can incite cross-reactivity with normal cell proteins. One of the most striking examples of this phenomenon is the release of protein arginine deaminases, or PADs, which convert arginine residues into various proteins to citrulline. This is relevant to RA because production of anti-citrulated protein antibodies has been observed in patients with severe progressive RA. In fact, it is believed that the presence of these antibodies defines a separate class of patients with RA. Intense stimulation of T cells or B cells, e.g. by bacterial endotoxin, can be so potent as to bypass the need for helper cells or co-stimulatory cells with the generation of autoimmunity. Likewise, an imbalance between pro-inflammatory and anti-inflammatory cytokines has been implicated in experimental autoimmunity. Finally, abnormal immune regulation because of failure to generate active Tregs has been associated with immune dysregulation and polyendocrinopathy. The inherited defect is failure to express FOXP3, a gene which is critical to the differentiation of Tregs. Again, we emphasize that multiple mechanisms can participate in development of autoimmunity, reflecting the multiple functions and controls of immune function. Indeed, Goodnow and others have proposed that, like the development of neoplasia, development of an autoimmune disease requires a series of abnormalities which ultimately results in the abnormal state, which no one defect sufficient in itself to precipitate the development of autoimmunity.